Okay, well, everyone, I hope everyone's doing all right. So this is very odd, as all of you know. I think that we've all had a bunch of odd experiences in the last several weeks of our life, in particular me right now talking to, I know all of you, but really to feel like just a screen. So anyway, today, I wanted to give you an opportunity to go ahead and ask questions. And so what I'm going to do, first of all, I'm going to go ahead and run it similar to what I've done in the past, two exam reviews that we've had. I'm going to go ahead and open this poll everywhere question or Q&A poll up. So I'm going to go there. Let's see if I can't get this started. Hopefully it's activate. Okay, so hopefully people can respond. I'm going to go ahead and run it through this poll everywhere, just because I think it's, quite honestly, it's easier for me to follow than chat, because if there's one question that a lot of you have, then I don't have to try and figure that out with chat. You can vote on this. So I'm going to go back to this. Let me see here, because I don't see anyone responding. I'm wondering if that poll is even active. So people on the chat, maybe if you could indicate on the chat, is it on Microsoft Teams, is that poll everywhere active for people? Okay, the poll is not working. Okay, so it's not active yet. So let me try it again. Hopefully this works. If not, then we'll just do it through the chat. I'm not sure why this wouldn't work. Should be activated now. Still not working. It doesn't appear. Oh, yes, there it is. So someone's able to get it to work. So it looks like. Okay, so we'll just go ahead and do it. Okay, so good. Questions are starting to come. So while people start to populate questions there, let me go ahead and talk a little bit also about the exam. So hopefully you've seen the announcements on Canvas about the exam. You're going to get 80 minutes to take this exam. I just feel like that's going to be more appropriate, considering that some of you might have slow internet or something along those lines. So you're going to get expanded time, which I think is nice. Hopefully all of you have good internet and nothing problematic happens. So and you can use the full 80 minutes. Of course, you don't need to. It'll be written just like the last exam. So it would be written to take in 50 minutes, although the online environment might make it a little bit different from that. So to make sure that you are ready to take the exam, remember I've posted that extra credit assignment. Hopefully you saw the announcement on that. I'm going to give you 5 points extra credit just for showing me that your computer is ready to take a poll everywhere exam. That's going to, I think, minimize the amount of problems that we are going to have on the exam, hopefully. That's the goal anyway. It's very possible that there are going to be problems even once you've fully demonstrated that your system's ready to go. Don't stress out about those. Go ahead and email me. I'll be on my email all day long. Or you can set up a direct chat with ProctorU as well. That's probably what we're going to have to do. So, but don't stress out. We'll try and get this figured out, okay? So just keep that in mind. Some other things about the exam that I haven't necessarily made an announcement on that I want you to all be aware of is that once you start the exam, your browser is going to go to full screen mode, and it will remain in full screen mode, and you won't be able to use tab browsing while it's going. So once you submit the exam, all you need to do is hit F11, and that will take you out of full screen mode, and then you'll be back to your normal computer function. I'm going to give you the ability to use two pieces of scratch paper, so obviously blank pieces of scratch paper. That way, if you do have, thoughts that you have to write down, then go ahead and use those scratch pieces of paper, okay? And then, one other thing, 80 minutes. So go to the bathroom before, so you can't have a bathroom break. It'll tag you for sure if you get up and go to the bathroom. So don't do that, please. go before, don't drink a lot of water, and be done with it, okay? So, all right, so let's go ahead, without further ado, go ahead and start looking at some of these questions. I'll look at this one at the very top. How much detail should we know about oxidative stress pathways? By the way, I'm going to try and write on this too. I know that for some of you that might be a little bit of lag, so I'll minimize the amount of time that I'm writing on this, but let me come back to the poll here. How much detail should we know about oxidative stress pathways? I would definitely know, expect you to know that you've got the matrix and you've got the cytosol, and you got two different ways really of producing the NADPH in that, the transhydrogenase in the matrix of the mitochondria. And then, of course, the pyruvate, or not the pyruvate, the pentose phosphate pathway, specifically, glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase are important enzymes in that. So those are all things that I would really expect you and want you to know. So I also, let me go ahead and I'm going to pull up here for us. Let me do this. There is this whole HIF1 slide here and here, okay, that I spent some time talking about. I would want you to know what HIF1 is. I would want you to know what its implications are for glycolysis and the expression of PDH kinase and what that's going to do in terms of flux through citric acid cycle and electron transport chain. So spend some time thinking about this slide and also in connection with, this slide and cancer cells. So we didn't spend as much time on the slide, so I wouldn't expect as much in this. Okay, but this is the connection to, of course, the pentose phosphate pathway and the possible anti-cancer therapeutics there as well. Okay, so keep those things in mind as you study for oxidative stress. Okay. Other things, certainly what is glutathione? I would expect you to know what glutathione is. What is the reduced form? What is the oxidized form? What reduces it? What oxidizes it? So the glutathione peroxidase, right, and those enzymes involved in that. So, okay. All right, okay. So hopefully that answers that question. If you have remaining questions, just go ahead and type them up. So the question is, the next question, the most popular question is, can you go over uncouplers again? So yes, I most certainly can go over uncouplers again. Let me go ahead and use this. Again, I'll try and use this sparingly, and I'll try and go somewhat slowly on this. Oh, awesome. Okay, there we go. I must have done something. But anyway, so with an uncoupler, we would have here, just go ahead and draw this. We'd have a P side and the inside. Of course, our P side is the intermembrane space, the inside being the matrix. Okay, so let me go ahead and just draw out here the matrix. Okay, and the P side would be essentially the intermembrane space. Okay, and then we would have here in this, let me get rid of that line here, draw just a big box here. Okay, we would have in this the electron transport chain that would represent complexes 1, 2, 3, and 4. Okay, so that's what's represented by that box. And then we would have what I have called the raddest molecule on the planet, okay, and known by mankind. We'd have that ATP synthase molecule, okay, or complex 5. All right, and what we know here, and what hopefully you understand, is that we've got protons that are being moved from the inside to the P side, okay, and then those protons come back through here, right, and they will go through this system, okay, and out through that ATP synthase, and in that process they will generate, they will generate ATP. Okay, all right, so that's the electron transport chain, that's ATP synthase. This is the coupled versions of these, okay, meaning that they're both dependent on one another, right? The electron transport, or I'm sorry, the ATP synthase is dependent upon electron transport chain to supply the protons in the p side to make ATP. The electron transport chain is dependent upon ATP synthase to essentially ensure that the proton gradient doesn't get too steep, is the term that I've used, okay? So that's the coupled system. Now, they can be uncoupled then, okay, by these molecules like DMP, okay, or there can be natural uncoupling as well. There can be uncoupler proteins, okay, but let's just go ahead and do this in an unnatural way. If DMP is here, what it can do is it can come through, okay, and it can actually grab onto one of these protons. So it'd be DMP, dinitrophenol, with a proton attached to it. Okay? And then that DMP can actually move into here and release the proton. So the proton will come off when it gets over here. Okay? And so essentially what we've done, or what this uncoupler has done, is it's moved a proton from the p side to the n side. And it has done so independently of ATP synthase. Right? So when everything is functioning in the cell, meaning that you have a functional ATP synthase, you have a functional electron transport chain, DMP or an uncoupler like it will actually reduce PO ratios, meaning that if your PO ratio is, let's say, 2.5 ATP per NADH, if you have DMP there, it might be something more like 2 or 1.5 or even 1 ATP per NADH. The reason being that not all of the protons being moved from the inside to the p side will be used to make ATP, because some of them will be grabbed through DMP and moved to the matrix of the mitochondria. Okay, and when they're released on that other side of the membrane, then heat will be used. Okay, so that's what uncoupling is. Now, the other aspect of this is you can actually inhibit. There's something called the ligomyosin that can inhibit ATP synthase. And if you inhibit ATP synthase, that should lead to, as long as your two processes are coupled, that should lead to a non-functional electron transport chain. Okay, something that that wouldn't then consume oxygen. However, if you inhibit ATP synthase and you use DMP, or you put DMP into the mitochondria at the same time, you can demonstrate that that electron transport chain can consume oxygen independently of a functional ATP synthase molecule. Okay, so that's uncoupling in a nutshell. Okay, hopefully that answers that question. Okay, all right. So, and if you honestly, if you want, by the way, if you're not understanding uncouplers, go ahead and unmute your mic for just a little bit and say, could you explain this to me? And I'm fine with that, okay? But you'll have to remute your mic again. So, I want you to have the freedom to do that as well, okay? Okay. Let's see here. So, can we go over the pentose phosphate pathway in relationship to oxidative stress? Yes, we can. So let me go ahead and come back here. Hopefully this is, I don't want to, again, rely too much on this, but I'm going to have to for these kind of questions. Hopefully it's working for everyone. There's not too drastic of a lag time for people. So what we have here is this. Okay, when we think about the pentose phosphate pathway, let me just go ahead and start here. Glucose 6-phosphate. Actually, we'll just draw glucose. Okay, we all know, hopefully by this time, we know what converts glucose to glucose 6-phosphate. The trapping reaction, hexokinase, does that. Okay, and then of course glucose 6-phosphate can go on, and the entire glycolytic pathway can go, and you get pyruvate down here. Okay, so that's glycolysis. Now, the connection to the pentose phosphate pathway is this, that glucose 6-phosphate, okay, can be converted in two primary steps to something called 6-phosphogluconate. Okay, and one of those steps here is one that we talked about in class, and I don't want to go over all the enzymes for this, okay? But one of the steps here takes this NADP+, I'm sorry, and makes NADPH, okay? That enzyme that does that is called glucose 6-phosphate dehydrogenase. There we go. Glucose 6-phosphate dehydrogenase. Okay, that's the name of that enzyme. Okay, and it makes this NADPH. Now, this can go on then. Okay, keep going on with this pentose phosphate pathway. That can make something called ribulose 5-phosphate. This out of the way. Okay, and that is going to make another NADPH. Okay. And the name of that enzyme, I'm not gonna write it down, but it would be 6-phosphogluconate dehydrogenase is what it is. I guess, well, okay, I'll just not write it down. 6-phosphogluconate dehydrogenase is the name of the enzyme that catalyzes this. I'm running out of room and all sorts of things, okay? But anyway, so the important point of this, though, in particular, what we went over in class is these NADPHs, okay? Because these NADPHs, what they're going to go on and do, and I've kind of not drawn this the way I should have, but I'm going to go ahead and do this. So they will take this glutathione, reduced glutathione, oxidized glutathione, GSSG, okay? And this can be converted to GSH. Okay, in fact, there would be two GSHs here, and that's reduced glutathione. But you need to do that, you need NADPH. Okay, and that'll make NADP+. Oops. And the name of that enzyme is glutathione reductase. Okay, now the question that was asked was, well, okay, well, what's the importance of all of this in regards to oxidative stress? Well, here's the importance of this. Okay, so oxidative stress, one of the markers of oxidative stress would be increased levels of hydrogen peroxide, and hydrogen peroxide can be converted to harmless water. Okay, by the use of glutathione, the reduced glutathione. And this is going to be kind of strange because it's, there we go. I'm kind of going in the wrong direction. I guess I should have drawn it this way. Sorry. Okay, do it this way, and we'll go hydrogen peroxide this way. There we go, NH2O. And glutathione will be oxidized in that, okay? And that enzyme is going to be glutathione peroxidase, okay? So that's this enzyme here. Glutathione peroxidase. Okay, sorry, my handwriting hasn't gotten any better because of the coronavirus. In fact, it looks like it's gotten worse. So sorry about that. But that's glutathione peroxidase, and it's going to use this glutathione, and that glutathione is made, okay, from the oxidized form of glutathione, and you have to have NADPH to do that. Well, what supplies NADPH? The pentose phosphate pathway, okay? Now, just so we understand, this hydrogen peroxide, by the way, okay, that hydrogen peroxide can be made from, right, superoxide radicals or something like that. For instance, just simply this superoxide radical, which is a reactive oxygen species, and take 2 protons here. And this is a spontaneous process. It's not a non-enzymatic spontaneous process, and you need an electron to do this. Okay, can make hydrogen peroxide, and okay, this would be a key marker of oxidative stress, right? And this can also go in this direction as well. So a removal of hydrogen peroxide from the cell is going to reduce oxidative stress, and to remove that hydrogen peroxide, as hopefully you see here, you need NADPH, and to get NADPH, in particular in the cytosol, you need the pentose phosphate pathway, okay? Now, we stopped here, just so we have some appreciation of this, we stopped here at this point in the pentose phosphate pathway. There are several more steps to the pentose phosphate pathway after ribulose 5-phosphate, okay? But this hopefully gives you some concept of that, okay? All right. Okay, so, and again, if you have questions, more questions about that, just let me know. Okay, so next question, do we need to know the specific path of the electrons through complexes of the electron transport chain? So I wouldn't expect you to know it as well as I had you diagram it for that group work assignment that you turned in on the 10th. So I would expect you to have some sense, though, of what redox centers are found in what complexes, okay, so that you can speak the language of the exam, okay? So, for instance, if I say a heme A group or a copper atom, then you should be thinking complex 4. If I talk about a Q cycle, you should be thinking to yourself complex 3, okay? If I say something like FMN, you should be thinking to yourself complex 1, okay? So I'm not going to have you diagram the electron transport chain and all the gory details that we went over in class, but certainly you need to be able to speak the language of the exam and have some familiarity with what is in each of the complexes. I would certainly expect you to know how many protons each of the complex pumps from N to P side. I would certainly expect you to know the order in which things are going through each of the complexes. That's kind of given by their name, so I think that that's pretty straightforward. I would expect you to be able to predict redox states of things based off of some form of inhibition or maybe a lack of oxygen or something like that. So you want to be able to predict redox states of various redox centers in the complexes. So those are things that I would hope that you would have some grasp of by the time you're ready to take the exam on Wednesday. Okay. How does rotation lead to ATP release in solution is our next question. Of course, that would be a reference to ATP synthase. And how does that occur? So I can't even draw something like that. But let me go ahead and do this. I did pulled these up. So let me go ahead and go to I think probably the best figure would be this one, okay, for this. So when we look at this, I'm going to ask you to focus on D here, okay? Oh, I can draw on that. That's nice. Okay, so focus on D there. And what you're looking at here would be, so if we come up here, put our eyeball right here, okay? Looking down at this lollipop type structure, that's the F1 component of ATP synthase. So when we put our eyeball right there, we're looking at it like this, okay? And right in the center of that, here, is that gamma shaft that we've talked about, okay, and that I talked about in class. That is actually what's rotating, okay? And it's rotating in a manner that it will have different contacts with the 3 different alpha and beta pairs. Okay, so we have, this would be alpha beta, that would be one pair. This would be alpha beta, that would be another pair. This would be alpha beta, that would be another pair. Okay, and the position of that gamma shaft is going to determine what conformation each of those alpha and beta pairs is in. Okay, if they're in the O conformation, then is not bound to ATP. Okay, if it's in the T conformation, you can see this here. So what I'm referring to, T conformation would be ATP bound, and then the L conformation would be ADP and inorganic phosphate bound. Okay, now this is what I'm going to say here. I'm going to draw like a little black dot right here. Okay, and that is one side of that central stalk or that gamma shaft. Wherever that black dot is pointed, that alpha and beta pair are in the O conformation. Okay, so they're lacking ATP. 120-degree rotation of that, the alpha and beta pair, so I will draw here a black square. Okay, and some of you are saying, I can't see that black square. I understand, I get it. So how about we draw a black X? That'll be easier. Okay, but that black X marks the side of the gamma shaft that has an interaction with the alpha and beta pair that allows it to be in the T conformation. Okay, and so then when we go from that 120 degree swing, if we can swing this black dot, that portion of the gamma shaft 120 degrees, then we can take what is normally in the T and force it into the O. Okay, so that will then eject ATP. Now, we didn't go over all the details of why that happens. Okay, in other words, when that gamma shaft happens, why does the alpha and beta pair reject, or why does it eject the ATP? And it turns out that portion of the gamma shaft actually runs into where ATP would be, the active site. Okay, so it actually just forces it out in that way. Okay, so what we talk about then, as we will look at ATP production by ATP synthase, is we kind of keep track of where this black dot is. Okay, and wherever that black dot is, 120 degrees away from that is an alpha and beta pair in the ATP bound conformation. Okay, and if you can rotate that, and you can do this, the C ring will rotate this, then it will force that alpha and beta pair from the T conformation to the O. So this will go from T to O once that rotation occurs. Now this will go from L to T, okay, and then this will go from O to L, okay, and our little X would have moved here, and our black dot, if we've gone 120 degrees, would have moved there. Okay, and then we move the black dot again, and it goes from, this one goes from T to O, okay, and this one goes from L to T, and then this one will go from O to L. Okay, and you can just keep going on and on and on in this way. Obviously, it's pretty cyclical from this point forward. Okay, so I don't know if that is what people are having or kind of confused about, but that's essentially the details of that. I will say this, on Canvas, I had failed to post the little cartoon of that YouTube cartoon of ATP synthase. So I went ahead and did that. So you can look at that. It's, I think, only like, I can't remember how long it is. It's like 5 to 10 minutes long or something like that. But go ahead and look at that. And it's actually, it actually has someone explaining everything that I just kind of explained. So look at that. That's in the modules, the lecture 9 modules. I think it's at the very bottom of it. Okay, so you can look at a video of ATP synthase and how rotation leads to ATP production. Okay. Okay. Yes, so on the exam, are we able to have a piece of paper to write on for pathways? Yes, so I mentioned this and you may have missed it, but I'm going to give you the ability to have 2 pieces of obviously blank scratch paper. Okay, so everyone can bring that and you can write whatever you want on it after the exam starts. Okay, and again, you have 80 minutes to take the exam, so yes. Okay. Can we go over the key topics for oxidative stress? So I've kind of already done that, I think. How much detail should we know about oxidative stress? So I've kind of already done that. So I'm going to go ahead and leave that one because I feel like I've already answered that one. Okay, PO ratios and how to interpret them. Yeah, so or maybe even how to calculate them and how to interpret them is something as well. So let's do this. PO ratio is just simply the amount of ATP that can and will be produced for every oxygen atom reduced. Okay, so if you think about it this way here, trying to think of a diagram that will allow us to do this. So let's go ahead and just draw a very simple diagram of the electron transport chain. Okay. Okay, so that's our electron transport chain. Hopefully you kind of capture the view of what we're doing here. We got 1, 2, 3, 4, and then we've got complex 5 or ATP synthase over here. Okay? Now, when we look at this, let me go ahead and do this. I'm going to say half O2. And that's going to become water. Okay? Half of an oxygen molecule is 1 oxygen atom. Okay? So that's going to be our O in PO, is this. Okay? Now, we know this, or hopefully you are starting to understand this. NADH enters into this cycle at complex 1 and becomes NAD+. Okay, there are 2 electrons here, so I'm just going to draw out 2 electrons. And what we did in class is that we traced those electron movement through here all the way to here. Okay, and it takes 2 electrons to reduce 1 oxygen atom, or half of an oxygen molecule. Okay, so that's right. That's why we trace 2 electrons through there, because the stoichiometry is per 1 oxygen atom. Okay, or half of an oxygen molecule. Okay, right. Now we also said this, that in this process, of course, there will be these protons that will be pumped. In this case, there's 4 protons here, an additional 4 protons here. Some of you might be able to hear my kids. We're all home. So they were crazy about a half hour ago. I had to go tell them that I'm going to be on this. So, but anyway, right. So 4 protons at complex 1, 4 protons through complex 3, and 2 protons through complex 4. Okay. All of those are moved by those 2 electrons moving through each of the complexes. And those 2 electrons find their origin here, at least for what we're talking about in NADH, and they find their termination in that oxygen. Of course, the origin of electrons really are from, carbons that come from either glucose or a lipid or an amino acid in the citric acid cycle and some other pathways, right? Beta oxidation for lipids or glycolysis for glucose or something to that effect, okay? All right, but nonetheless, we've got these protons that have been moved from N to P side, okay? And there's a total then, if you were to add all these up, 4, 4, and 2, that's equal to 10 protons. Okay? Well, let's just say here, this is where it becomes really fun. Let's say this is a 9 subunit C ring. Okay, well, that's a 9 subunit C ring. Then it will take a total of 4 protons to make 1 ATP. Okay, and so I'm going to draw this over here. This is going to get the PI in, the inorganic phosphate in. Okay, and we have ADP. Oops. The PI plus ADP can be used to make ATP. Okay, we need 3 of these protons to go through here. I won't draw it the way it's going to do it, but we need 3 protons for there, and then we need an additional proton here. Okay, and that is the total number of protons to make 1 ATP, 4 protons. Okay, well, the P and PO ratio is that ATP, so PO. Okay, and it took 4 protons to make an ATP molecule if our C ring was 9 subunits. Okay, well, the PO ratio, what it is essentially saying is this. Okay, well, to reduce 1 oxygen atom, we have 10 protons, okay, that are going to be moved from the inside to the P side. It takes 4 of those to make 1 ATP, so we can make then 2.5 ATP per oxygen reduced, okay, because 10 divided by 4 is equal to 2.5 ATP. I don't know if any of you can hear my dog, but he just walked in the room and is like scratching his back on a door or something like that. I don't know what he's doing. But anyway, all right, so 2.5 ATP per NADH. Okay, and that is our PO ratio. Okay. So hopefully all that made sense. The reason I divided this by 4 is because it took 4 to make the ATP. Okay. So with FAD, of course, it would be a different number of protons that come through, or not FAD, maybe succinate, if you want to think about it that way. It would be 6 protons, okay, divided by the 4, and that would be 1.5 ATP per FADH2. Okay, so that's PO ratios. Okay, now there was another part of this question besides just, it said, where is it? PO ratios and how to interpret them. Okay, so I think that hopefully I answered your question with how to interpret them. This just simply means that for every NADH that's produced, okay, this here means for every NADH that's produced in a particular catabolic pathway, that will lead to 2.5 ATP. So if we're to think about, let me just go ahead and do this, because I've had some questions on this. Let's go ahead and do the whole enchilada, if you will. Okay. So if we go glucose to pyruvate, this is going to make 2 NADH. Okay. This is going to go into the mitochondria. I won't draw our divisions here, but this is going to be converted to acetyl-CoA. That's going to make an additional 2 NADH. Okay, this is going to, of course, as we know, it's going to combine with oxoacetate. Okay, and the whole citric acid cycle is going to run. There's going to be 2 CO2 made here. By the way, this makes it CO2 as well, 2 of them. Okay, but this is also going to make 3 NADH. And this will also make 2 FADs. Okay, so when we start to think about the total number of ATP that can be made from a glucose, going from glucose to essentially carbon dioxide and water, right? We have to think about those two NADH, these two NADH, these three NADH, and these two FADH2s, right? These, okay, will lead to 2.5 ATP per NADH, assuming our C ring is 9 subunits. Okay, so then you have to just do the arithmetic. We have 2, 4, and then 7 NADH that are made from that, and then this would lead to 1.5 ATP per FADH2. Okay, so we can start to then, assuming that all these electrons that are coming off of glucose are going to go into oxidative phosphorylation, we can start to calculate the total number of ATP that are going to be made by, in this case, the catabolism of glucose. Okay. Now we left off here substrate level phosphorylation. Of course, glycolysis makes 2 ATP directly. Okay, so this will make 2 ATP, and this makes 2 ATP directly as well. So we'd have to include that in our totals. Okay, and this would be net 2 ATP. Okay, right. So that's how you use those to interpret them. Now, of course, if C ring changes, that will change the PO ratio. If you have DMP, that's going to change the PO ratio, or an uncoupler or something to that effect, right? So there's lots of things that will change that, but that's what PO ratio is. Okay. And by the way, I did run into this in one of my office hours. This is a PO ratio for FADH2. This is the PO ratio for NADH. Okay, so we calculate them separately. Okay, let's come back here. Hopefully that answered that question. Let's see here. We've got some more time. How many ATP equivalents will be produced if the citric acid cycle ran all the way to ATP synthesis? Oh, so that actually dovetails really nicely with what we just asked, or what we were just going through. So let me come back here. Okay. So when you talk about just the citric acid cycle, let me go ahead and do this. The question is, how many ATP can be made from this pathway? Well, so we would have 3 ATP per NADH here, so this would lead to 7.5 ATP. Oops. Okay, and then this would lead to here, there would be 2 of them, there would be 3 there. Okay, because there's, actually, no, I'm sorry. Oh, gosh, I'm going to give myself a red check minus. I just realized something here. Red check minus, forgive me, my goodness. This would have been, for the last question with glucose, this would have been 6 NADH. So red check minus for me, my goodness. I'm hoping that everyone was like, yeah, that wasn't making sense. Darn you, it was confusing enough in the first place. Now it's worse. Okay, so I'm sorry about that. All right, so it should have been 6. I should have doubled it. 3 is for 1 turn of the cycle. Okay, now we are talking about, in this question, our current question, how many ATP equivalents will be produced if the citric acid cycle ran all the way to ATP synthesis. So in that current question, you only go through the cycle 1 time. Okay, and this would be, go back to the 3 here. Okay, and that would be 7.5. This would be 1. And there would be 1.5 ATP produced there. Add those up, and that's a total of 9 ATP, plus this would be 1 ATP because it's only going 1 turn. All right, so 10 ATP per cycle, okay, per turn of the cycle. And that would include substrate level phosphorylation and oxidative phosphorylation, okay? You double that when you're talking about glucose because glucose breaks down into 2-acetyl-CoAs, and so you have to run this 2 times. Okay, sorry about that. Forgive me. Okay. All right. Let's see here. How many ATP? Okay, on the exam we'll be able to have a piece of paper. We've already talked about that. Does coenzyme Q accept 2 protons at a time, and if so, where? No, usually it's going to accept 1 proton at a time because it goes through radical form. And it usually accepts those protons from the inside. Okay, if you look at, for instance, complex 1, the end of complex 1, it has to get an electron from an iron sulfur center, and that long chain of iron sulfur centers. And so it's going to accept one electron, and then when it gets that electron, it'll accept a single proton as well, okay, from the inside. The chemistry just allows it. We're not going to go over the chemistry details for that, nor are we going to. But it would be one proton. And then if you look at the end of complex 2, it's the same thing. I have iron sulfur centers that hand off an electron one at a time to coenzyme Q, and it picks up one proton at a time, again from the inside. Okay. All right, how much detail do we need to know about the electron carriers in each of the complexes in the electron transport chain? I would know, if you think about it this way, I would expect you to understand that how many electrons each of them carries, right? So you have 2 electron carriers or 1 electron carrier, so you want to know that. I would expect you to know, where essentially in the cycle what they would be found. Or not the cycle, but the electron transport chain. So like I said, I think earlier at the very beginning of the review, if you hear heme A3, you should be thinking to yourself complex 4. You should also know, generally speaking, redox potentials are increasing as we go from complex 1 and 2 to complex 3 to complex 4. So they get higher in reduction potential. Beyond that, and there's not too much to know. Okay. Okay, so how much detail do we need to know about the electron carriers? Okay, we just did that. Let me make sure that we haven't had something come up here. Can we go over to the pentose phosphate? Do we need to know specific? Okay, how's rotation? Okay, we talked about all those things, so just making sure no one's voting on those things. Can we go over the Keen cycle? I don't know what that means. The Keen cycle, I'm maybe the citric acid cycle, is that what's being asked? So I'm going to leave that one. Maybe if you had that question, there's only 2 of you, go ahead and email me and clarify that for me. Can we talk a bit about the regulation of PDH complex and the citric acid cycle? Yeah, we can. So let's go ahead and maybe make this the last one. We'll see. Let's see if we can't do this. So let's go. I'm trying to figure out what's the best way to do this. Let's go ahead and go here. Not there. Not that one. Let's go ahead and talk about this. Okay. So in terms of the citric acid cycle and pyruvate dehydrogenase complex, okay, this would be the slide that has kind of not all of, but much of the regulation in one slide, okay? Most of this is a discussion on allostery, okay? And the key point that I would want to make with this and have made in class is this, that NADH, okay, is an allosteric inhibitor or competitive inhibitor of citrate synthase, of alkyl-ketoglutarate dehydrogenase complex, and also the pyruvate dehydrogenase complex. Okay, now what I would expect you to go on and think about is, well, what would lead to higher levels of NADH? Okay, and that is primarily a question in regards to how rapidly is NADH being used by the electron transport chain, and what would cause that to slow down. Anytime the electron transport chain starts to slow down, NADH will start to accumulate. Okay, now that can be due to a lack of oxygen, which can lead to oxidative stress. That can be due to a lack of ADP, which can actually lead to oxidative stress. Okay, that can be due to some form of inhibition, unnatural inhibition of electron transport chain, which can lead to increased NADH. Okay, in all of those cases, though, I mentioned oxidative stress because there will be this abundance of electrons in the electron transport chain that can then be used, not purposely, but will be used to make reactive oxygen species. Okay, in all of those cases, because of how damaging oxidative stress can be to the cell, it's very important to stop making NADH, and it's very important to slow flux through the citric acid cycle so that oxidative stress doesn't become an enormous problem. Okay, now incidentally, with oxidative stress, NADH levels can lead to greater production of NADPH. Okay, so if I were to come here, see if I can't come back to this. Here, I have NADH. If those levels are going up in the cell, they're going to feed back and inhibit the citric acid cycle, but they can also be used by this transhydrogenase to make NADPH, and NADPH can then be used by glutathione reductase, as we talked about, and then that can be used, the glutathione that's produced from this can be used by glutathione peroxidase to make water. Okay, so in those cases, accumulation of NADH will actually be a self-corrective problem. It can actually lead to a reduction in reactive oxygen species. Okay, all right. So when we think about the regulation of these, though, coming back to this, that becomes a really key factor. Now, the other really key factor of this is this. Come here. We did this before spring break. It's been a while. It seems like forever, actually. But one of the things that we talked about was this enzyme PDH kinase. So PDH kinase is the full name of it. And PDH kinase will phosphorylate pyruvate dehydrogenase and inhibit it. Okay? Now, what activates it? Well, NADH activates it, or acetyl-CoA activates it. Okay, so if levels of NADH are rising in the cell, that will activate PDH kinase, which will then phosphorylate pyruvate dehydrogenase and inhibit it. Okay, not only that, but those will also, NADH will also directly inhibit pyruvate dehydrogenase complex in the first place as well. Okay, so now this PDH kinase, okay, becomes a really important point in some of our discussions on oxidative stress. We come here, come to this, and it's this enzyme. Okay, oxidative stress, okay, can be caused by hypoxia, low oxygen, paradoxically it can be, and that will lead to the activity of this thing called HIF1, hypoxia-induced factor. Hypoxia-induced factor is a transcription factor, okay, with genetic term that will essentially, it's a protein that will essentially lead to genetic expression of different genes. And one of the genes that activates, okay, or leads to higher levels of expression is PDH kinase. So when there's low levels of oxygen, which will lead to the production of reactive oxygen species, then PDH kinase will be expressed in higher levels, and that will then inactivate pyruvate dehydrogenase complex, and that will then slow the citric acid cycle and slow the respiratory chain or electron transport chain, and that will then slow the amount of electrons there and reduce reactive oxygen species. At the same time, the same factor, HIF1, can lead to increased levels of glycolytic enzymes, a glucose transporter, and lactate dehydrogenase, okay, which will lead to greater flux through glycolysis. And by the way, it's not shown here. Not sure really why it's not shown on the slide, but glucose can be converted to, remember, glucose 6-phosphate, and that can be used in the pentose phosphate pathway, which makes NADPH. Okay, so then NADPH can be used to reduce oxidative stress. All right, so that's some of the connections that I'm hoping you'll make in terms of oxidative stress and the regulation of the citric acid cycle, the regulation of the pyruvate dehydrogenase complex. Okay, so I'm glad that we got to that question, because I think that it answers some of the questions that we've had about oxidative stress as well. Okay, this gave me a little bit more form to go ahead and mention that. Okay. Okay, so let's see here. I think that's sufficient. I think we've all been here long enough. So yeah, we've got to pretty much most of these questions. So if you have like burning questions on your mind that just didn't get answered in this, go ahead and email me those questions, and we go from there. Okay, so I think that we got over the majority of people's concerns. Okay, so So take care of everyone. So I honestly, I do miss you. So I wish I could see you all, but I can't. So let's do this. Is it 3 and 8H per 1 acetyl-CoA? Okay. So I'll look through these. I'm just going to let all of you go. I'll look through these comments here, and see if I missed anything. Okay, on this. Okay. All right. Have a good rest of your day, everyone. And in Colorado, it's snowy weather, so I can't really say enjoy the weather unless you just really love the snow in April. So, okay. All right. Take care, everyone.